Hello, this is Tim Sandle back with you with another video looking at all things to do with clean rooms. And for this week's short video we're going to have a look at spores and the problem of spores posed in aseptic areas and clean rooms in general. I'm going to be looking at bacterial spores, or what are known as endospores. They're called endospores because they're produced from within the bacterial cell. Now, the first thing about spores is that not all species, and in fact only a very narrow range of bacterial species, can produce spores. So organisms like Clostridia, which has species that can cause botulism, and Bacillus, which is found in soil. Now, these organisms under unfavourable conditions can engage in a survival tactic. So essentially they're producing a copy of their cytoplasm and their genetic material, and they're encasing that in a three-layered structure and expelling that. And in essence that's a complete copy of the original vegetative cell, so it's kind of very similar to a clone. And what's amazing about spores is that they can remain in this state for incredibly long periods of time, theoretically hundreds of years, and then reproduce when the conditions are again favourable. They sense whether there's water present, or they sense whether there's nutrients present, and then they enter again into the vegetative state. Now, these spores are incredibly hardy. They can survive very, very dry conditions, ultraviolet light, levels of radiation, and even heat to a degree. So this makes them very problematic, and particularly when they come to disinfection and cleaning as well. So what do these spores look like? Well, here's an electron micrograph of Bacillus spores, and these are taken on rubber, and you can see the spherical structures. Now, spores are very, very difficult to destroy. As I've said, they can survive starvation, and they actually survive most types of disinfectants. It's only those disinfectants that have sporicidal properties. And also spores can be spread. They won't by themselves move, but they can be picked up by people, picked up on trolley wheels, picked up on cardboard, and so on. And the fact I was saying about spores lasting a long time, there's an interesting experiment in a university in Sweden where they have a jar of alcohol, jar of ethanol, and that contains bacterial spores. And once a year they take out a sample and they're seeing whether the spores can germinate, form the vegetative cells. And they've been doing this for around 150 years, and the spores to this date are still capable of coming back to life. So just shows how hardy those spores are. Now, what does a spore sort of look like, and why is it so tough? Well, the spore can be thought of as a bit like a walnut. So you've got this really hard outer casing, and that's supported by an enzyme called superoxide dismutase, and that gives it a degree of resistance. And then inside the spore are these various layers. So we have the exosporium, and that acts as a really tough barrier to disinfectants. It's quite hard to get through that layer. And then even if disinfectant gets through, then the cortex and the inner membrane act to prevent the diffusion of disinfectants. So you've kind of got these multiple layers that are enhancing the resistance of the spore. So it becomes very hard to break, even for sporicidal disinfectants, which is why things like contact times and the right concentration are absolutely important. Now, how do spores end up getting into the pharmaceutical facility? Well, there's numerous sources. There's bags, there's boxes, pallets, scrubbing machines, shoes, and so on. But probably one of the big risks is the materials coming from warehouse into production facility, particularly when weather's wet, particularly where there's cardboard. So it's really important that deliveries come If they're wet, they're allowed to dry in some kind of airlock or annex, and then all the outer layers are removed. So items should ideally always be triple wrapped, so you can take off a layer before it even gets anywhere near the clean room. And of course, as we've heard before, cardboard has no place in a clean room. It is full of spores and is a really horrible substance to have anywhere near the clean room environment. We also have concerns from damaged surfaces. So where there's any kind of cracks, these become niches or reservoirs for spores, and often a good survival point. So, and particularly where the inner walls are exposed, where we've got things like plasterboard and so on, they are really quite dangerous substances. And also we have things that come in, so like shoes are really bad, which is why we always have to have captive shoes, even in the first stage changing rooms, and why we have to get rid of all our outdoor clothing. And of course, even if we do good disinfection practices and other good practices, it's more likely at floor level there's a risk of spores, so which is why during processing we never ever pick up objects from the floor. We always wait for the end of the work shift. So we can address spore problems by good cleaning. We need to clean effectively in order to allow disinfectants to work, and then using special disinfectants called sporicides, and always applying that in the form of saturated wipes or spraying or putting the disinfectant onto the surface via a bucket and mop, and then always wiping. So just applying a disinfectant is not overly effective. We need to wipe, and if we start to see it drying out before the contact time has elapsed, then apply again. And then also we have protection from well-designed clean rooms, good air change rates, keeping pressure differentials, and making sure all the surfaces are cleanable. And then also with ourselves, as I said before, practicing good gowning. And we can also minimise the risk by having good practices like using multi-pack items and so on. Okay, so this brings this presentation to an end. So what we've looked at is bacterial spores, why they're so hardy, how they form, how they have a degree of resistance to disinfectants, so we really need to make sure we're applying those contact times, and some of the places they might be coming from, particularly incoming materials into the facility, and we have to be really good at transfer disinfection, and we always apply a sporicide at each step, and we always take off a layer at each step, going from clean but non-classified areas into grade Ds or Cs, and then transfer into B, and then before transfer into grade A. Okay, so that's the end of the video. I'm Tim Sandle. Good luck with the rest of your day, and until next time, I'll be back with you again. So, cheery bye.